So our next speaker is Dr. Santosh Kumar from Agriculture and Agri-Food Canada in Brandon. Santosh is a research scientist at the Brandon Research and Development Centre. He completed his master's degree at the Indian Agricultural Research Institute in New Delhi, India, before moving to Canada for his PhD program. As a PhD student at the University of Manitoba, Santosh worked on barley physiology and genetics, and he lives in Brandon. Thanks, Santosh.
Well, do I have this mic? Yeah, okay. Can you hear me at the back? Yeah, okay, perfect. Well, I did my PhD from this department, and I did my postdoc as well. So I spent 10 years in these buildings. I was scared of this room because I wrote a few exams in this room. So, where are my slides? There we go. And all right, so I'm a wheat breeder, and I'm also the team lead for breeding at Brandon, and I also act as station director from time to time. That's why very few hairs left on my head. Importance of wheat. Studies have shown that since Neanderthals, every human evolutionary step has consumed one or the other form of wheat. So I hope you all eat a lot of wheat. Otherwise, okay, so global production of wheat is about 750 million tons. Canadian average is about 35 million tons. We produce a fair bit of wheat. Out of that, $82.9 billion worth of wheat is exported. In Canadian context, it's about $8.2 billion Canadian. So it's a very high export value crop. Out of these wheats, Canada Western Red Spring type class of wheat is the predominant, and it's grown at about 70% of the acreage in Western Canada. So it is accountable at a very large acreage. Out of those acreage, 80% of the wheat acreage is under Agriculture and Agri-Food Canada varieties. So AFC is a major player in variety development that is also taken up by the farmers to grow on their fields. An average return to investment for every dollar invested is about $22, and that's a very conservative number. It ranges between 20 to 35%. These are avenues where wheat is used. The one thing that surprised me that wheat is used in cosmetic products too. I wouldn't know anything about it, but who do use cosmetics, they'll tell you that it's a very important ingredient. Status of wheat in Canada is quite large. It accounts for about $42.7 billion in revenue. To the people who are directly involved in wheat, that's the full-time equivalents, and that's 215,000 people who are involved in wheat directly or indirectly. And that translates into about $16.8 billion in wages. So it's not just a crop, it's an economy itself. So when we talk about breeding and development of cultivars, why is that even important? You know, there are many cultivars that are present and farmers can choose to grow it forever. But what about the evolving pathogens? What about the export requirements that change continuously? What about the new technologies that are coming into the varieties? A new technology can be implemented into the new cultivars, and that's when it makes the farm scale impact. For example, if we have biological nitrification inhibition, a new trait that is coming into our wheat varieties, that has the potential to be grown at millions of acres and can reduce the greenhouse gas emissions. So your new variety becomes the vehicle to take that new technology at a farm scale where it will have an impact. Otherwise, the research and the technology has no impact if it's not grown. And that's why breeding is so important. Our focus areas are for yield, disease, and quality. The Canada Western Red Spring has optimum levels of quality for export market. It is excelling in the yield, and it has optimum disease resistance to all major priority diseases in the prairies. We are looking at research for trait introgression, just like I mentioned, the biological nitrification inhibition, or midge tolerance, or FHB tolerance. So we continuously look for newer traits and then introgress it into varieties. And the upstream research is focused in areas of genetics, genomics, and physiology. And I'll talk a little bit about that in my future slides. These are the AFC sites where we do research. So I'm locating at Brandon, and we primarily develop the Canada Western Red Spring type wheat. We have stations at Ottawa and Charlottetown. For us, these are the early warning stations for FHB, new FHB races that are coming into Canada. That gives us enough time, by the time it reaches from here to here, that we have some resistance in our varieties that we are releasing. So this is our outpost, kind of. As we go towards the west, we have swift current, which is more in the drier regions. So if we are looking at incorporating drought tolerance, tolerance to less water, less moisture, and high nitrogen use efficiency, that's what is happening at swift current. Brandon has decent amount of rainfall, but we have lots of diseases, and that stays our focus in this area here. As we move towards Lethbridge, we are looking at high yielding class, the soft white class. We all love cookies and cakes. That's where the soft white wheat goes. So that's the research station that develops varieties for those. And we are looking at Beaver Lodge. This region, with global climate change, there will be a significant amount of acreage that will open up in this region. As you can see that it is very narrow right now, but we expect that there will be lots of acreage here. So we need to develop varieties that are suited to this region. Canada is a large, large country with a huge geographical area, and varieties developed in the southern region do not do well up. So we have to have focus where we can develop varieties that are short season, but long day crops. And that's why the focus here in Beaver Lodge. I look after the program at Beaver Lodge and Brandon. Agronomic priorities, you are very familiar. Second priority is yield, and the third one is yield. They want yield. They want yield in the CWRS class because that translates into money for them. We need to have appropriate maturity. You heard the last talk from Claire, that if we let it drag too long, then the farmers would be harvesting in snow. Nobody wants that. So we have to keep that maturity within range. The longer you leave a plant in the field, the more photosynthesis it can do and the greater the yield, but we can't let it go too far. So the yield is kind of dependent on maturity. Lodging resistance, if the farmer has to harvest a lodged crop, he loses time. He uses more gas on his combine. That's not desirable. So we can't let the plants lodge. Shorter straw, no farmer wants to deal with straw, entangled straw in their combines catching on fire. It's a pain to do, and it slows them down again, consuming a lot more fuel. So we have to have shorter straw. Pre-harvest sprouting is directly related to quality, and test weight is an indicator of milling. So a grain has to be milled to be translated into product. So if the test weight is not optimal, there is a loss in the milling yield. And that's why these are the key priorities from an agronomic point of view, and we have to account for that. Then the diseases, there are 5 priority 1 diseases, the 3 rusts, leaf rust, stem rust, stripe rust. Then we have the most important fusarium head blight, because it produces toxin in the grains. And then common bunt, which is not prevalent in the eastern parts of Canada, but it is quite prevalent in a susceptible variety out in the West. So it is important priority. Then we also look at loose mud and leaf spot because that has become a problem for the farmers in the recent years. Even though these are not required for registration of a variety, we do want to incorporate and provide a good package to the farmers. Finally, the quality. The CWRS wheat is exported to 82 countries annually from Canada, giving about $8 billion every year, and about 24% of the GDP. So it is a massive crop for export, and we need to have the export quality that the importers want, not just what we produce. So we constantly talk to the Canadian Grain Commission and look at the parameters that they gave us to meet. And that changes from time to time. For example, a few years ago, a gluten strength was needed to be adjusted, and it needed to be stronger. We can't change a breeding program really quickly, but within 2 to 3 years, we had a good number of varieties for the farmers to grow that met the new requirements. And that's why breeding needs to continuously evolve. So we need 13.5 or above protein in wheat. We need the optimal gluten strength, and we need to have the proper color in the noodles. Can't just make good noodles. It should have good color too. Japanese are very particular about this. So those are all factored into breeding program. So when we develop a variety, we're not just looking at agronomy, we're not just looking at disease, we're not just looking at quality, we are trying to develop a package with all 3 different characteristics. Finally, how do we do breeding? That's a complicated thing, but I'll simplify. We make the crosses, P1 and P2, and they would be bringing newer traits, sometimes complementary traits, and then we add a 3rd parent to it, because why stop at 2? If we have the technology, let's bring in another parent and then make a good population. So we grow about 300,000. If you have attended annual day at Brandon, you'll see this expanse of 300,000 individually planted F2s. And that's because if you look at Genetics 101, F2 is the generation where all your traits have the potential to show their effect. After that, it starts to segregate and it doesn't show you the traits. But there is a limit. If you look at the table on the left here, if you have just 1 gene, 2 F2 plants are enough to show you the traits that are different. If you have 10 genes, you need 1,000 plants. If you have 20, then the number grows exponentially, and so on. We can't do that much work. So we limit ourselves to what we can do with the people we have and the land resources that we have available. These plants are inoculated with rust and FHB. So there is the inoculation requirement, inoculum requirement, inoculation time period, and then looking at 300,000 plants. We do this every year. All of this every year. This is just one of the generations. So it's very time consuming. That's why we limit ourselves to 300,000. Then we select roughly 6,000 out of 300,000. And that gets sent to New Zealand for winter nursery. We make selections again, and out of those 6,000, about 1,500 come back. These are put into small scale trials just to make sure that the traits we started with are still there. We didn't lose any new traits that we incorporated. Once that happens, then it's sent back to New Zealand again for seed increase and selection, and then it comes into advanced scale trials and registration trial. That's where we look at, would this line perform at a larger geographical area? Or is it just at Brandon that it performs well? So these trials here emphasize the need for genetic plasticity and also the performance plasticity of the lines over a larger geographical area, different soil types, different rainfall regime, different fertilizer input, and all of that. And finally, if we are lucky, at the end of this, we will have one variety out of all of that. maybe. Once the variety is developed, it goes through a lot of regulatory processes, and Canadian Food Inspection Agency registers the variety after the support from the Prairie Grain Development Committee. And once it is registered, then the seed industry takes it over. The seed industry takes the line and produces select seeds, then foundation, then registered, and then certified seeds. These certified seeds are the ones that goes to the farmer for growing in their fields. While this is happening, provincial trials do happen, and that becomes the source of Manitoba Seed Guide, where you look at the performance of lines while this is happening. So there are many things that happen, and this is a very short summary of that. This picture just shows you half seeds after crossing as they develop in the spikes. Finally, the breeding technologies that we use, adaptation sites. Seems like it's just a site where we grow yield plots. But these are the ones that tell us whether that line will be acclimatized to that location, is acclimatized to that location, will it perform there or not? So it is one of the most important tools in breeding, the registration trialing sites. Specialized nurseries, FHP nursery, rust nursery, pre-harvest sprouting, testing nursery, those are very important to give us the data set that are required before we register the varieties. Marker-assisted selections. A lot of things we can see in the field, but a lot of things we can't. If there is a new disease, for example, UG99 family of rusts, it's not in Canada yet, but it may come, it will come at some point of time. We need to have resistance before it comes. We have markers that that show us that the line will be resistant to that race of rust. And that's why markers are important. When we have pyramiding of genes, multiple FHP genes, multiple leaf rust genes, when we see a resistant plant, we don't know what is causing that resistance. So markers allow us to track which gene is providing the resistance out of the 5 that may be present in the plant. So that's why markers are important for gene pyramiding. When we showed you the complex cross, the P1, P2, and P3, that's a complex cross, and we use markers to now narrow down the required or favorable alleles very early in the generation. And if we do that and remove plants that don't have the favorable alleles, they don't take up space in the land and in the breeding pipeline. So it's very important to have markers for the traits that we are interested in, especially when we are doing complex crosses. Winter nurseries, they save us a lot of time, because if we have to grow those lines every year in Canada, that will just double the time required to develop a variety. So winter nurseries are very important. And double haploids as well, because in a traditional selfing pipeline, wheat is a self-pollinated crop, so we call it the selfing pipeline. It takes about 8 generations to reach uniformity and homozygosity. In double haploids, you do that in 2. So it saves a lot of time. Finally, the markers we routinely use. There are multiple markers for leaf rust, stem rust, stripe rust, FHP, pre-harvest sprouting as well. And recently there was a line that we released, and it turned out to be really okay for PHS, but one bad year and it started to PHS, and we tested it, whether it had any of the markers or any of the genes, and it had none. So most years the lines will be okay, but if you don't have the gene, you don't have the effect in a poor year. And that's why it's important to have these markers to test out the lines, even if the trait is not visible during the growing season. We could miss it for 11 years, and the 12th year it will show up. That's why it's important to have those tested. Various markers for yield. These are for bunt, and you can see some bunted kernels there. It has a fishy smell. If you have bunt susceptibility and infection in your crop, you drive by it and you'll smell fishy, rotten fish. No farmer wants that, so we do want to have that resistance. Midge is an important insect pest of wheat, and it can cause yield losses up to 15%. There is just one source of resistance. We don't have any other gene globally that provides resistance to that wheat midge. So we have midge-tolerant lines, and there is a midge stewardship where we grow the resistant line with a 10% refuge. And what it means is that you have 90% of your resistant lines, and you add 10% of a susceptible line so that the insect has something to feed on. And it reduces the natural selection, and the insect doesn't develop resistance to this gene. So it's an industry in itself now, the midge-tolerant lines. And then finally, for harvest indexed, we have the dwarfing gene, RST1. We are trying to move away from RST1 because it causes FHB susceptibility in wheat, and we are trying to move on to RST24, still causing the dwarfings, but a different source of dwarfism. Oh, this should be like this. Okay. Talking about newer technologies, how much time do we have?
5 minutes.
Okay. So in a traditional breeding pipeline, like I explained, we do the crosses. We either use double haploids or the inbreds, and they go into small-scale trials, large-scale trials, and release of varieties. We have brought it down from 12 to 8 using the DHs and the winter nursery. That's a significant gain because newer varieties make newer crossing parents, and then they make varieties again. So better varieties in the hands of the farmers sooner. That's 4 years sooner. That increases the transgressive segregation gains per year. But a newer technology where we are moving away from the markers because, yes, if there are traits that are major gene effect traits, you can use marker-assisted selection. But what if a gene is needing assistance from many other genes? And that's when genomic selection, because you're now assaying many other genes in the population, not just the major effect gene, that's where genomic selection comes from. In this case, you have a training population. Training population is genotyped and phenotyped. So all your parents that are going into crosses become part of the training population. And training population trains the model based on the genotype and the phenotype. Once the model is developed, you can minimize your phenotyping needs because that's the most time-consuming and most expensive part of breeding. We go through the model based on the training population data, and we just do large-scale trials for adaptation and release of varieties. That also takes about 8 years. But it allows you to account for a lot more genetic variability within your population and select lines that have unique gene combinations, sometimes minor alleles. And that's an advantage. This is helping us get better lines that have better package. You can have lines with high agronomic value, high disease value, or high quality value, but this is allowing us to bring the package together in a better way. And then phenomics. This is still in infancy for breeding. Yes, there are lots of work done in very particular traits, but for breeding, where we mostly rely on a very small megapixel camera that is our eyes and a supercomputer that is our brain, this is something that we have to consider very carefully because there are lots of data generated. Lots of different things that we can capture that we don't usually see in the field. For example, canopy temperature. How green is the plant? What is the population density of a certain type of cross? All of those can be assayed using different sensors, cameras, but we haven't really incorporated into our breeding program at a very large scale because of the data analysis challenges. In breeding programs, a lot of the people are willing to go out in the field and look at the plants, but they don't want to sit on the computer and analyze data. So it is a challenge for us. Finally, these are the outcome of the breeding pipeline. And I'll start from the bottom, the most recent ones. How many of you have seen the 2026 Manitoba Seed Guide? Except you. Okay, so in the CWRS class wheat, it's a shout out for that seed guide as well. You'll see reverse and ahead. So the most common varieties these days is AAC Brandon. And whenever we are doing comparisons, we are comparing it to Brandon. So AAC reverse is about 11% higher yielding than Brandon. In the last 5-6 years, we tried our best and we could never reach more than 8% of Brandon. This is the line you can see in the seed guide, is about 11% higher yielding than Brandon. Ahead is about 8% higher yielding than Brandon, but has a better disease package. So these were just released. If we go backwards, Craven was about 7% higher yielding than Brandon. Walker is about 6% higher yielding than Brandon. So you can see that as we go from years, 23, 24, 25, we are increasing the yield. And this being a big jump in that yield, 4% in a year. So with that, breeding is not done in isolation. Breeding requires a talented set of people and people who are passionate about what they do, because it's very, very time consuming and hard on the body. And this is the team that does it. So big shout out to them. And with that, nothing really moves forward in research till there are many people giving money for that. These are the funding partners. Canadian Wheat Research Coalition is supporting us with 5-year research grants. Alberta Grains, Ask Wheat, Manitoba Crop Alliance are important funders for us. And we are supported by the seed companies as well in different capacities. So big shout out to them. Thank you very much. No questions?
Okay. Thank you very much.
Thank you. Thank you. That's not needed.